Here is your essay generation prompt. Please begin writing. Begin. Essay. The year 2008 witnessed the official recognition of Harald Hausen's profound contribution to human health through the award of the Nobel Prize in Physiology or Medicine. His identification of human papillomavirus, or HPV, as the causative agent in cervical cancer represented a paradigm shift in cancer research and prevention. This essay undertakes a critical evaluation of Hausen's body of work culminating in that prize-winning discovery, meticulously examining the claims, evidence and arguments presented concerning the link between HPV and cervical carcinoma. The analysis will proceed section by section, scrutinizing the internal logic and supporting data while also acknowledging the broader scientific context that was shaped by this groundbreaking risk. Section 1. Initial Hypotheses and the Search for Viral. Etiology. Hausen's initial foray into the field of cancer virology was driven by a skepticism of the prevailing dogma that herpes simplex virus type 2, or HSV2, was the primary etiological agent in cervical cancer. This skepticism, rooted in epidemiological inconsistencies and limited experimental evidence, spurred him to explore alternative viral candidates. His early work involved the investigation of various papillomaviruses, initially those associated with benign skin warts. The claim that papillomaviruses, specifically those with a propensity for infecting mucosal tissues, might be implicated in cervical carcinogenesis represented a bold departure from established thinking. The initial challenge lay in identifying and isolating viral DNA within cervical tumor cells, a task greatly complicated by the low viral load and the integration of viral DNA into the host cell genome. The section's crucial aspect resides in the claim that the viral presence can be detected at all, even when not producing actively replicating virus. This early phase laid the groundwork for the development of molecular techniques necessary to detect and characterize HPV within cervical cancer samples, methods that would later prove crucial in establishing the causal link. One should consider the intellectual climate. The dominance of herpesvirus research meant securing funding and resources for papillomavirus research was a significant hurdle. This environment undoubtedly influenced the early stages of hypothesis formation and experimental design. Section 2. Development of hybridization techniques and viral identification. A significant advancement in Hausen's research came with the refinement of in situ hybridization techniques, enabling the detection of specific HPV DNA sequences within cervical cancer tissue. This involved creating complementary DNA probes against known HPV types and using them to identify the presence of matching sequences within tumor cells. The central claim within this section is the specificity of these techniques. They needed to reliably differentiate between various HPV types and, crucially, avoid false positives from non-HPV sources. This required meticulous optimization of hybridization conditions and rigorous controls. The success of this approach led to the identification of novel HPV types, subsequently designated HPV-16 and HPV-18, within a substantial proportion of cervical cancer samples. The finding that specific HPV types, rather than a generic papillomavirus infection, were consistently associated with cervical tumors provided powerful evidence supporting their role in carcinogenesis. It is important to acknowledge the laborious and painstaking nature of these early experiments. Each hybridization required meticulous preparation and careful analysis, a far cry from the high-throughput sequencing technologies available today. The development of these fundamental techniques formed the bedrock for all subsequent research in the field. Section 3. Viral integration and cellular transformation. The observation that HPV DNA was often integrated into the host cell genome within cervical cancer cells, rather than existing as extrachosomal episomes as is common in benign warts, provided a crucial clue regarding the mechanism of HPV-mediated carcinogenesis. The claim here is that this integration isn't merely a passenger event, but rather a critical step in the transformation process. Integrated HBV DNA often disrupted or altered the expression of cellular genes involved in cell cycle regulation and apoptosis, contributing to uncontrolled cell growth. This section of research focused on mapping the integration sites of HBV DNA within the human genome and correlating these integration events with changes in cellular gene expression. The identification of key viral oncogenes, such as E6 and E7, whose expression was enhanced upon integration, provided a molecular explanation for HBV's transforming potential. This line of inquiry also addressed a significant question: why only a small percentage of HBV infections progress to cancer? The answer, in part, lies in the specific integration sites and the resulting disruption or aberrant expression of cellular genes. Subtle nuances in viral integration patterns could determine the ultimate fate of an infected cell. Section 4. The role of E6 and E7 oncoproteins. The discovery and characterization of the E6 and E7 oncoproteins represented a major breakthrough in understanding the molecular mechanisms of HBV-induced carcinogenesis. The central claim here is that these proteins directly interfere with key cellular regulatory pathways, driving uncontrolled cell proliferation and preventing programmed cell death. E6 was shown to bind to and promote the degradation of p53, a critical tumor suppressor protein responsible for maintaining genomic integrity and initiating apoptosis in response to DNA damage. E7, on the other hand, was found to bind to and inactivate the retinoblastoma protein, or PRB, another crucial regulator of the cell cycle. By disabling these key tumor suppressor proteins, E6 and E7 effectively remove the brakes on cell proliferation, allowing cells with damaged DNA to replicate uncontrollably. The detailed elucidation of these molecular interactions provided strong evidence for the direct causal role of HBV in cervical cancer. Moreover, the identification of E6 and E7 as key oncogenes opened up new avenues for therapeutic intervention, focusing on targeting these proteins or their downstream effects. It's worth noting the elegance with which these two viral proteins subvert cellular machinery. This highlights the evolutionary pressure on viruses to develop sophisticated mechanisms for manipulating host cells to their own advantage. Section 5. Epidemiological evidence and cofactors in carcinogenesis. While the molecular evidence for HBV's role in cervical cancer was compelling, robust epidemiological data was essential to solidify the causal link. This section focuses on the claim that HBV infection is a necessary, though not sufficient, condition for the development of cervical cancer. Numerous epidemiological studies demonstrated a strong correlation between HBV infection, particularly with high-risk types like HBV16 and HBV18, and the incidence of cervical cancer worldwide. These studies also identified various cofactors that could increase the risk of HBV-related cervical cancer, including smoking, co-infection with other sexually transmitted infections, and compromised immune function. The understanding that HBV infection alone is insufficient to cause cancer is crucial. The interplay between viral factors, host genetics, and environmental exposures determines the likelihood of disease progression. Further, careful analysis of the epidemiological data revealed the variability in HBV prevalence and cervical cancer incidence across different populations. This highlighted the importance of public health initiatives aimed at promoting HBV vaccination and cervical cancer screening, particularly in regions with limited access to healthcare resources. This section also addresses confounding factors and biases in epidemiological research, ensuring a rigorous assessment of the evidence. Section 6. Animal models and experimental validation. The development of animal models to study HBV-induced carcinogenesis proved challenging but ultimately crucial for validating the causal link between HBV and cervical cancer. The claim here is that these models, while imperfect, can recapitulate key aspects of the disease process and provide valuable insights into the mechanisms of HPV-mediated transformation. Initially, researchers struggled to infect standard laboratory animals with human HPV. However, the development of transgenic mice expressing HPV oncogenes, as well as the use of xenograft models in immunocompromised mice, allowed for the study of HPV-induced tumor development in vivo. These models demonstrated that expression of E6 and E7 was sufficient to induce precancerous lesions and, in some cases, invasive cervical cancer. While these models did not perfectly replicate the human disease, they provided valuable experimental support for the role of HPV in carcinogenesis and facilitated the testing of potential therapeutic interventions. This section reflects on the limitations of relying only on in vitro studies. Establishing causality requires demonstrating that HPV can induce cancer in a living organism, even if the model is a simplified representation of the human. Section 7. The development of HPV vaccines. The unequivocal establishment of HPV as the causative agent in cervical cancer paved the way for the development of prophylactic HPV vaccines. The central claim regarding these vaccines is their ability to prevent HPV infection and, consequently, to significantly reduce the incidence of cervical cancer. These vaccines, typically containing virus-like particles, or VLP, that mimic the structure of the HPV capsid but lack viral DNA, elicit a strong antibody response that neutralizes the virus and prevents infection. Clinical trials have demonstrated the remarkable efficacy of HPV vaccines in preventing infection with high-risk HPV types and reducing the incidence of precancerous cervical lesions. The introduction of HPV vaccines represents a landmark achievement in cancer prevention, offering the potential to dramatically reduce the burden of cervical cancer worldwide. This section acknowledges the challenges associated with vaccine development, including the need to ensure long-term efficacy and safety, as well as addressing concerns about vaccine hesitancy and equitable access. It is also a testament to the power of basic research in translating scientific discoveries into tangible benefits for public health. The journey from identifying HPV to developing effective vaccines is a prime example of how fundamental research can have a profound impact on human lives. Section 8. Screening and early detection strategies. While HPV vaccines offer a promise of preventing cervical cancer, screening programs remain essential for early detection and treatment of precancerous lesions, particularly in populations with limited access to vaccination. The claim here is that cytological screening, such as the Papillomavirus test or Pap smear and HPV DNA testing, can effectively identify women at increased risk of developing cervical cancer and allow for timely intervention. The Pap smear involves microscopic examination of cervical cells to detect abnormalities that may indicate precancerous changes. HPV DNA testing, on the other hand, detect the presence of high-risk HPV types within cervical cells. The combination of these two screening methods provides a comprehensive approach to cervical cancer prevention. Women identified as having precancerous lesions can undergo treatment, such as cryotherapy or loop electrosurgical excision procedure, or LEAP, to remove the abnormal cells and prevent progression to invasive cancer. This section acknowledges the limitations of screening programs, including false negative results and the need for regular follow-up. Implementing effective screening programs requires infrastructure, trained personnel, and community engagement to ensure that women have access to and participate in these life-saving interventions. This emphasizes the importance of integrating prevention strategies with treatment options in a comprehensive approach to cervical cancer control. Section 9. Global health implications and future directions. The discovery of HPV as the causative agent in cervical cancer has had profound global health implications, particularly for developing countries where cervical cancer remains a leading cause of death among women. The claim here is that the combination of HPV vaccination and screening programs offers the potential to eliminate cervical cancer as a major public health problem worldwide. Achieving this goal requires addressing various challenges, including ensuring equitable access to vaccines and screening services, developing affordable and effective screening technologies, and overcoming cultural and logistical barriers to implementation. Future research efforts should focus on improving sensitivity and specificity of screening methods, developing therapeutic vaccines to treat existing HPV infections, and understanding the mechanisms by which HPV causes cancer in specific populations. International collaboration and resource mobilization are essential to translate scientific advances into tangible benefits for women around the world. This section also considers the broader implications of Hausen's work for cancer research in general, highlighting the importance of investigating infectious agents as potential causes of other cancers. The success in identifying HPV as the cause of cervical cancer has inspired researchers to explore the role of viruses and other microbes in the development of various other malignancies. Section 10 critiques and subsequent refinements of the HPV model. While Hausen's work revolutionized our understanding of cervical cancer, it was not without its critics and subsequent refinements. Some argued that HPV infection alone cannot fully explain the development of cervical cancer and that other factors, such as host genetics and environmental influences, play a significant role. This section addresses the complexity of the carcinogenic process and acknowledges the limitations of a purely viral-centric view. Subsequent research has focused on identifying genetic variations that predispose individuals to HPV-related cervical cancer, as well as exploring the role of the microbiome and other environmental exposures in influencing disease progression. Furthermore, the mechanisms by which HPV evades the immune system and establishes persistent infections have been the subject of intense investigation. These refinements have led to a more nuanced understanding of the interplay between HPV, the host, and the environment in cervical carcinogenesis. This section emphasizes that scientific progress is an iterative process, with initial findings being challenged, validated, and refined over time. The ongoing research into HPV and cervical cancer reflects the dynamic nature of science and a continuous pursuit of a more complete understanding of this complex disease. Conclusion. Harald Hausen's groundbreaking discovery of human papillomavirus as the causative agent in cervical cancer represents a watershed moment in the history of cancer research. His meticulous investigation, from initial skepticism of prevailing theories to the development of sophisticated molecular techniques, laid the foundation for a paradigm shift in our understanding of carcinogenesis. The identification of HPV as a necessary, though not sufficient, condition for cervical cancer paved the way for the development of prophylactic vaccines and improved screening programs, offering the potential to dramatically reduce the global burden of this deadly disease. While subsequent research has refined our understanding of the complex interplay between HPV, the host, and the environment in cervical carcinogenesis, Hausen's seminal work remains the cornerstone of our knowledge. His legacy extends beyond cervical cancer, inspiring investigation into the role of infectious agents in other malignancies. Hausen's contribution exemplifies the transformative power of basic research and its profound impact on human health, illustrating the potential of scientific inquiry to alleviate suffering and improve lives across the globe. His work stands as a testament to the enduring value of rigorous scientific investigation and its capacity to reshape our understanding of the world.